Good morning, students. This is Murthy sir. In the last class, we discussed about raisin experiment and the potato cup experiment. Today, we will discuss about another two interesting experiments related to plasma membrane. Students, already you know, cell membrane is selectively permeable. It allows solvent to pass through it, and it allows only selected solutes to pass through it. Students, the aim of the experiment is to understand the movement of materials in and out of cells through cell membrane. Students, the materials required for this experiment are two beakers, one funnel, one filter paper, retort stand, sugar, wheat or rice flour, 500 ml plastic bottle, cotton cloth, tincture iodine. Students, here you can see the apparatus, retort stand, beaker, funnel, filter paper, and the solution that you are pouring into it. Now let us see the procedure. Prepare wheat or rice powder solution in a beaker by adding 1 teaspoon of powder in 100 ml of water. Add a drop of tincture iodine to the solution. Students, tincture iodine is iodine mixed with alcohol. Pour the solution into the funnel. Students, you know, iodine is generally used in laboratory for conformation of starch. It will be dark violet in color. Students, let us see the observation. The funnel, which contains filter paper, allows water, dissolved sugar, and tincture iodine to pass through it. All these materials will reach the beaker present below, but it will not allow wheat or rice flour to pass through it. This wheat or rice flour will remain in the funnel. Friends, now let us see the conclusion of the experiment. Cell membrane is selectively permeable. That is, it allows solvent and only selected solutes to pass through it. Students, now let us move on to the next experiment, which is related to the process osmosis. Students, the aim of the experiment is to observe endosmosis and exosmosis in a D-shell egg. The apparatus required for this experiment are 3 beakers, petri dish, salt, dilute HCl, 2 equal size raw eggs, 1 tablespoon, a strip of paper for measurement, a cloth to wipe. Students, when you place raw egg in a beaker containing dilute HCl, this dilute HCl being an acid, it can dissolve the eggshell, which is basically made up of calcium carbonate. Students, the procedure of the experiment is keep 2 raw eggs in dilute HCl for 4 to 5 hours. Take out deshelled eggs with tablespoon and wash them in tap water. Students, due to the action of hydrochloric acid, the eggshell will dissolve and you will get a deshelled egg, which is required for this experiment. Students, now we have to measure the size of the egg with a long strip of paper. Prepare concentrated salt solution in a beaker. Place 1 deshelled egg in salt solution and the other in tap water. Students, just like the raisin experiment, the deshelled egg which is placed in salt solution, it undergoes exosmosis and it shrinks. And another deshelled egg which is placed in tap water, it swells due to endosmosis. Students, when you observe the size of eggs which are placed in both beakers, you will find a decrease in the size of the egg which is placed in salt solution, and you will find an increase in the size of the egg which is placed in tap water. This is due to exosmosis and endosmosis. The egg placed in concentrated salt solution shrinks due to exosmosis. The egg placed in tap water swells due to endosmosis. The conclusion of the experiment is in osmosis, water or solvent moves from less concentrated solution to more concentrated solution by crossing semi-permeable membrane. Students, already you know this osmosis practically it takes place in living systems. You learned some examples. The absorption of water by root hairs from the soil, the opening and closing of stomata, which is very important for exchange of gases and transpiration, and the filtration of impurities from blood in the nephrons of human kidneys. All these are practical examples which you have to remember.